So it's my honor to present at your excellence meeting. Special thanks to Jill, she makes this possible. My research interests are in the mechanism of general anesthesia. General anesthetics are the only tools that allows to turn human into an object. But they are the only tools that allow to do so reversibly. And the big question is how reversible those actions are. So mounted evidence indicate that traces of anesthetic actions that remain after anesthesia withdrawal can develop in profound abnormalities later in life. So they can induce side effects. And obviously, this becomes an important problem. Importance is more evident when we consider number of patients that are exposed to general anesthesia. You may know that daily in the United States, 60,000 surgeries are performed. And according to these statistical studies, 1.5 million babies are exposed to general anesthesia during the first year of their lives annually in the United States. So these are unbelievably high numbers because they make more than 1 in 4 newborns in the United States. Majority of human retrospective epidemiological studies found cognitive deficiencies and symptoms of ADHD in those who were exposed to general anesthesia early in life. And the US FDA, based primarily on animal data, issued a recommendation to avoid, whenever possible, anesthesia in pregnant women and children younger than 3 years of age. The exact content of this recommendation can be questioned now, but this is another indication of wide recognition of importance of this problem. So how anesthetics induce these adverse effects is not entirely clear. Today, I'll present some of our findings, but because many of you do not deal with this problem on a daily basis, so I will make a brief introduction to this topic before I start with actual experimental findings. And in this field, in the field of adverse effects of general anesthetics, there is general agreement that these effects are age-dependent. They are more prominent in very young. This idea is especially attractive if we take into consideration that major molecular substrate for action of majority of general anesthetics to induce the state of general anesthesia, GABA type A receptors, they function very differently in immature brain. So you know that GABA is major inhibitory neurotransmitter in the brain. It causes inhibition by activating chloride current through GABA type A receptor channels, shown here. So which one is this one? Yeah. And in majority of mature neurons, in most of neurons, not in all, the transmembrane gradient for chloride is such. So the transmembrane gradient for chloride is such that the equilibrium potential for chloride is almost the same or a little more positive than the resting membrane potential. You may remember that the equilibrium potential for a given ion is a potential at which the net conductance for that ion is equal to 0. So this is the potential to which a given conductance attempts to shift the membrane potential. It cannot shift the membrane potential to values more negative than its equilibrium potential. This means for the GABA conductance that GABA cannot hyperpolarize membrane potential. So GABA causes inhibition of neuronal activity not by hyperpolarizing membrane potential, but by shunting more excitatory conductances. The essence of shunting inhibition is described by Ohm's law. If we reduce the membrane resistance, this is what we do when we increase the chloride current, greater excitatory currents are needed to shift the resting membrane potential to the threshold of action potential, to excite neurons. GABAergic anesthetics do not alter the transmembrane gradient of fluoride, at least acutely. At the time of anesthesia, they increase fluoride conductance. Therefore, general anesthetics also cause inhibition of neuronal activity by shunting more excitatory conductances. And the situation is quite different in immature brain, especially in the rostral areas of the brain, such as the cortex, hippocampus, hypothalamus. Many neurons in these areas have high intracellular concentration of fluoride because of high expression of the NKC1 and low expression of the KCC2. So in these neurons, the equilibrium potential for fluoride can be near or even more positive than the threshold of action potential. This means that in these neurons, GABA causes not inhibition of neuronal activity, but strong depolarization and even excitation. This depolarization is sufficient to activate calcium influxes through voltage-operated calcium channels and NMDA subtype or glutamate receptors. And those calcium influxes are central for regulation of many developmental processes. So obviously, any agent that can either up or down regulate GABA receptor signaling would alter those calcium influxes and will affect developmental processes in the brain. General anesthetics are one of them. And we found if you administer, for example, GABAergic anesthetics such as Sevoflurane to neonatal rats, they cause epileptic seizures and also multifold increases in major stress hormone, corticosterone. Normal brain development assumes that GABA receptor signal in transit from excitatory to inhibitory. In rodents, this takes place during the second postnatal week. In humans, presumably by the end of year 1. And it occurs because of developmental increase in expression of KCC2, impairment in KCC2 expression, and impairment in transition in GABA receptor signal into inhibitory have been linked to many developmental psychiatric disorders. We have found that if you administer GABAergic anesthetics to neonatal rats, such as Sevoflurane, they can induce not only seizures, increase in corticosterone levels, but they can induce profound, lasting impairment in expression of KCC2. Similar changes can be found in these disorders. So adult rats neonatally exposed to Sevoflurane have reduced KCC2 expression in the brain. They exhibit exacerbated hypothalamic pituitary adrenal axis response to stress. Their major stress response system function abnormally, and they exhibit a number of behavioral deficiencies. You may know that very similar abnormalities can be induced by excessive perinatal stress, suggesting that these two phenomena may share similar mediating mechanism. You may know also that perinatal stress can induce inter- and transgenerational effects. Therefore, we tested hypothesis that if we administer Sevoflurane to neonatal rats, we can see abnormalities not only in the exposed animals, but also in their unexposed progeny. To test this hypothesis, we exposed postnatal day 5 rats to anesthesia with Sevoflurane. We used concentration 2.1%. This concentration is sufficient to block, to depress the writing reflex. The writing reflex is frequently used as animal model to study efficacies of anesthetics to induce consciousness to depress consciousness in humans. But this level of anesthesia is not sufficient to conduct surgery. So this level of anesthesia is not sufficient to induce immobility. When the exposed animals achieved age of 3 months, we use them as breeders to produce offspring of all possible combinations of control and the exposed animals. The offspring, labeled F1 generation, parents belong to F0 generation. The offspring were never exposed to general anesthesia, And they were tested in a number of behavioral paradigms followed by gene expression studies. So the first behavioral paradigm was done on postnatal day 60. It was the elevated plasmase in behavioral neuroscience. This test is used to assess anxiety, stress-type behavior. If animals spend shorter time in open arms as opposed to enclosed arms, this is considered as an indicator of increased anxiety, stress-type behavior. And to our surprise, so I need to mention that, yes, we found significant abnormalities in offspring, but only in males, not in females. Females offspring exhibited trends, but no significant changes. Therefore, I will focus mostly on data obtained from F1 males. So to our surprise, F1 males of exposed father, control mother, not offspring of both exposed parents, spent significantly shorter time in open arms of the EPM. The next behavioral test was done 10 days later. It was the prepulse inhibition of acoustic startle response. Few words about this test. So you know that intense, unexpected stimuli, such as sound, visual, or tactile, can cause startle response in humans and animals. It's mediated by a relatively simple neuronal circuitry in the brainstem. Here you can see a start response recorded in rats using this equipment in response to this sound stimulus. So start response is a defensive response. When startle stimulus is preceded by a weak subthreshold repulse, then response to start stimulus is profoundly reduced. This phenomenon is called prepulse inhibition of startle. It's used to measure ability of the brain to filter non-essential information, sensory, motor, cognitive information. PPI of startle is mediated by much more complex neuronal circuitries, which are not completely understood at this time. You can see from the diagram that multiple neurotransmitter systems, including GABA, are involved in modulation of PPI of startle. PPI of startle is impaired in human patients with many psychiatric disorders, especially with schizophrenia. In schizophrenia patients, there is strong correlation between extent of impairment in PPI of startle and negative symptoms of schizophrenia, cognitive symptoms. So the importance of PPI of startle is in that in contrast almost to all other behavioral paradigms used in laboratory animals, this technique or this method can be used in humans and animals using almost exactly the same protocol. So it can be easily replicated between species. And this may facilitate translational research. We found that, again, male offspring of exposed father, control mother, exhibited deficiencies in PPI of startle at 2 prepulse intensities, 3 and 6 decibel above background. So you can see that adult offspring of exposed parents exhibited deficiencies that can be found in schizophrenia patients. The next behavioral test was done between postnatal days 79 and 85, the Morris-Watermaze behavioral paradigm. You may know that behavior during this test relies primarily on hippocampal function. This test consists of 2 parts. During part 1, animals are trained to locate a hidden platform. With each training session, they spend shorter time, and this is reflected in learning curve. And you can see in this graph that there was a trend, but no significant treatment group effect on learning during the Morris-Watermaze test. Animals, offspring of both exposed parents, this gray line, exhibited trends to spend more time to locate the platform, but the difference was not big enough to achieve statistical significance. During the last day of this test, platform is removed, and the time that animals spent in the target quadrant where the platform was located is recorded as indicator of spatial memory. And you can see that offspring of both exposed parents, that gray bar, spend significantly shorter time in the target quadrant, suggesting that their spatial memory was impaired. When these animals were 3 months of age, brain tissue was collected to look into gene expression levels. We looked specifically at NKC1 and KC2 chloride transporters because we had extensive prior data on involvement of these transporters in effect of general anesthetics in the exposed animals. And we found that F1 males of exposed father, control mother, had significantly increased expression of NKC1 in the paraventricular nucleus of the hypothalamus, central brain region for the hypothalamic pituitary adrenal axis. KCC2 was impaired in the hypothalamus in offspring of all combinations of exposed parents. But in the hippocampus, the most affected group were F1 males of both exposed parents. So those were the only group that exhibited reduced expression of KCC2 in the hippocampus. And you may remember that this was the only group that exhibited deficiencies in the hippocampal function-based Morris bottom-based behavior test. And obviously, neonatal parental anesthesia affects behavior and gene expression in offspring. Therefore, in order to get an insight in potential mechanism, we used bisulfate sequencing to look at DNA methylation profile in KCC2. And so we looked in promoter region. We designed primers based on data on sequence published in this study. They have shown that if mice are exposed to BPA, then the KCC2 is hypermethylated. No, not the KCC2, but KCC2 in cortical neurons of the offspring is hypermethylated and expression is depressed. So we found that in sperm of F0 males neonatally exposed to sevoflurane, these are adult animals, they were 5 months of age. They were exposed to sevoflurane at or on postnatal day 5. You can see that the KCC2 promoter was hypermethylated. In F0 females exposed to sevoflurane, there was a trend of increase in KCC2 methylation, but no significant effect. There was no group effect in hypothalamus and hippocampus of F1 females, but in F1 males, there were significant group effects. F1 males of all combinations of exposed parents had significantly hypermethylated KCC2 in hypothalamus. F1 males of exposed father, control mother had KCC2 methylated greater than all other groups, and those males were that exhibited deficiencies in EPM and PPI. In the hippocampus, the most affected groups were F1 males of both exposed parents. So those F1 males that had significantly reduced KCC2 expression in the hippocampus and exhibited deficiencies in the hippocampal function-based Morris bottom-based behavior. At the beginning of this presentation, I mentioned that there is a dogma in this field that the effects, these adverse effects are age-dependent. Adults are considered resilient to such effects. But from the literature data, we knew that germ cells can be susceptible to epigenetic reprogramming initiated by different environmental factors throughout the lifespan. So we tested the hypothesis that if we expose young adult rats to sevoflurane, then we will not see any changes in the exposed animals, but we may see some effect in the offspring. So to test this hypothesis, we exposed young adult rats for anesthesia with sevoflurane on 3 alternating days, starting on postnatal day 56. And then 25 days later, we use them as the breeders to produce offspring, again, of all combinations of control and exposed parents. We run a number of behavioral paradigms, gene expression studies, bisulfite sequencing, and we were surprised to find that intergenerational effects were very similar, not exactly the same, but very similar to those that were induced by neonatal exposure to sevoflurane. Only F1 males were affected. KCC2 expression was reduced. They exhibited deficiencies in these behavioral paradigms. And we found that in F1 male hypothalamus and hippocampus, KCC2 was hypermethylated, and it was hypermethylated also in sperm and ovarian tissue. So not all oocytes, but ovarian tissue. So altogether, these results suggest that anesthetics like sevoflurane may induce intergenerational effects, and these intergenerational effects may occur when parents are exposed to general anesthetics over a very broad spectrum of ages, from neonates to young adults. In these studies, we look specifically at expression and modulation of KCC2, again, because we have a number of publications showing that KCC2 is involved in mediation of adverse effects of anesthesia in the exposed animals. We realized that it's unlikely that KCC2 is the only gene that is involved in mediation of this intergenerational effect. And this possibility is supported by our preliminary data, where we applied RRBS to sperm of adult male rats that were neonatally exposed to sevoflurane. And you can see that more than 2,000 differentially methylated DNA regions were in males that were exposed to sevoflurane when they were compared to control. Most of these sites were hypomethylated. These orange and green dots represent p-values of significantly differentially methylated DNA regions in males that were exposed to sevoflurane. So most likely these intergenerational effects are mediated by very complex mechanisms. There is a lot of work to be done here. And another interesting observation is that in young adult anesthesia study, so our hypothesis was that the exposed animals will not be affected. But contrary to that hypothesis, we found that sevoflurane induced profound increase in corticosterone levels in males and females at the time of anesthesia. But long term, only males were affected. They exhibited profound neuroendocrine abnormalities, behavioral deficiencies. But you may remember that germ cells were modified both in males and in females. And males and females passed abnormalities to their male offspring. So this brings us to a possibility that offspring are more susceptible to side effect of parental anesthesia with sevoflurane than the exposed parents themselves. So parents may be normal, they don't exhibit any changes in phenotype, but their offspring may be affected at multiple levels. Okay, this is all our research team, our funding support. Again, thank you very much for inviting me, and thank you for your patience.